Combining scientific experience with entrepreneurial accumulation, Dr. Jena champions the translation of idea into impactful innovations. So today we will talk about case studies, startups, entrepreneurship, and innovation in medical technology. So I will request Dr. Nivedita Jena to deliver the his presentation, her presentation to the participant. Thank you.
Thank you, Dr. Mahanti, for your kind introduction. I'm very honored to be a part of this FDP program that's going on for 6 days. And I congratulate Trident group of institutions to conduct such a thoughtful program for faculty development. So can I share my screen?
Yes, ma'am, you can share your screen. Ma'am, please share your screen.
Yes. Just give me a minute, ma'am. There are too many windows open. I'll just. Is it visible, ma'am?
Yes, ma'am, it is visible.
All right. Can I make it full screen?
Yes, ma'am. Take it full screen. Yes, ma'am. It's fine, ma'am.
All right. Thank you so much. So for my understanding, I think they're all faculties teaching from engineering background who are listening to me. Is that right, ma'am?
Yes, ma'am. Some participants are from academic background and some are from research scholar and some are from industry also.
All right. Okay. So I have a mixed cohort. So please feel free to interrupt me during my presentations if you have questions. How I have compiled my talk today is bringing what is required in the biotech medical innovations in the current state in India right now. Where are we heading towards? What are the startup ecosystem available with us to take our innovations to next level? How faculties, how researchers or industry people can strengthen this ecosystem and bring meaningful innovations to the society is what my talk will revolve around. As we know, biomedical innovations, focus mainly on affordable, scalable, and context-oriented design for low-resource health settings, mainly. So when you look at biomedical technologies, it's a wide range of healthcare products that you see, from devices that we use in our house for measuring glucose, for measuring PPs, to measure the oxygen contents. These are all health tech devices that the technology has provided to the society that can ease our lives to learn more about our health. So this covers main of the diagnostic, prevention, monitoring, treatment, and elevation of any diseases. The main crux of medical technology innovations are clinical outcomes, less invasive processes, and shorter recovery time. This is the main reason why the innovations are happening in today's world. Now, if you look at the process of innovation, where does it begin? How does it begin? So I completely believe, understanding the clinical needs. And doctors are the first person to know if the clinical need is there or not, because that's where they have the direct contact with the patients, right? They have direct contact with the treatments they are giving to the patient's complaint. So they are the ones who will understand where is the gap and what needs to be done. So we as a faculty, we as a technologist, we as a researcher need to talk more often to the doctors in certain areas of your focus that you like, that or that you want to venture into. They will be able to get us the problem gaps, which is which is the first step for any process innovations. So once we know the clinical need, okay, that's when we can think of an idea. Ideation and working on a correct problem statement is more important. And I think that is the more, one has to take more time to understand if the problem that you are selecting to innovate or you are selecting to find a solution is really a problem worth enough to devote your time. If that problem is impacting 1,000 people, 10,000 people, or a masses of lakh or more than that, that is where these questions needs to be asked to decide if the idea that you are working on or that you have thought in your mind is worth pursuing or not. Now, once you clear that step, okay, you move on to proof of concept. You figure out methods and methodologies how to create a proof of concept of an idea. Then you work on proof of feasibility, followed by proof of value. And then once your prototype is ready and a market viable product is ready, then you enter into clinical trials by approaching test license agencies. So CPSEO, CE, ISO are all regulatory bodies we need to clear before we need to approach to understand their requirement and then design our clinical trials in that way, so that the validation of our solution is more meaningful before we get approval and launch. Once the launch is done, it is all ready for clinical use and standard care. But once launch is done and people have started using it, there is another survey all of us have to do, is the post survey of marketing, where we understand what is the gap that we have left while innovating the right, can bring in the newer version of the same device or diagnostic or an assay that we are developing for a clinical need. So in a crux, any innovation that we are working on follows the same cycle of or the process. So first the clinical, then the market business, regulatory, and focus is on technology bringing to the society. Now, ideation and implementation needs to go together to bring an outcome that can have a business benefit. So innovation is only 1% of ideation, 9% of evaluation and polishing that we work, and 90% of implementation, right? So a friend of mine always says, our mind gets 60,000 ideas per day, but how many get executed? It's based on us, based on our priority, based on our wish, passion, how we are creating all of those factors in to decide if these ideas are implementable or not. And the same holds true for any idea. All right. So ideas, we get plenty. We need to figure out it should be worth spending time and it should be worth implementing. And when implemented, it should be able to give us benefit. And this is the crux of complete. As a faculty or coming from academics, we always say, research is all about using money to create knowledge. All right. Innovation is using knowledge to create money. This is the striking difference between research and innovation. All right. Now, moving forward, we have seen a lot of medical devices or implants that are being available. And many of our startup companies and individuals have come forward and done a lot of innovations in creating devices for patient aids. For example, hearing aids, pacemakers, we have been hearing this since long. Similarly, a lot of innovations have gone into implant. A lot of materials have been, newer materials have been introduced as implants so that the acceptability has increased. Okay, that is also a sector which is increasing at the rate of 25% CAGR. And we have stents. Stents are also a major component of devices that are continuously used in medical field, which also is being evolving over the years with different kind of more adaptable materials coming into the market. Equipments and instruments, for our healthcare systems, we have a lot of evolved MRI machines, CT scanners, ultrasound machines, dental drills, dental chairs. Now we have also chairs that can just are movable. We can just ask dental chairs. Moving dental clinics have also come up. So people are designing new kind of machineries for movable healthcare systems and portable healthcare system that can be taken to every household and can be used. Of course, consumable and disposables are another sector which is very huge and large, which comprises of disposable plastic syringes, needles, blood bags, IV fluid, and many other that we regularly see. This is a very big market. Also, this market, people are now looking at replacing plastic, the single-use plastic in this market, which is a very, very important aspect of innovation that Atmanirbhar Bharat, under that umbrella, all the ministries are trying to promote finding out solutions in this category. Now, the key growth drivers of biomedical innovation industry in India are the faster upgradation of existing technology and the new product innovations. The ones that we have been using a lot of machineries that is coming from either US, Europe, or other countries. But now people are really working towards creating your own technologies, creating your own machines that can enable and make us more self-reliant. And then a lot of awareness is helping us to create advancement in medical technology, and everybody is coming together to put their skill set and ready to take a risk. The change in mindset is very important for the growth of any industry per se. The same is also visualized in medical technology landscapes. We are also looking at improving delivery of healthcare. There are a lot of apps, AI integrated devices that people are working on, that is increasing trend of technologies, and then reducing the time of healthcare interventions. So all of that is major force, or people are looking at innovation. And there's also changing patient profiles, increasing life expectancy and aging population. Now, there are a lot of non-communicable diseases also, which needs intervention from healthcare. Always now with the increase in, most of us are able to afford, purchasing power has been increased. So all of these are major, thrust for demanding new innovations into the market. Having Blinkit coming to your doorstep and then delivering your food, it's an ease in life. Similarly, Apollo Pharmacy coming with medicine to your doorstep, that's also a device. And if healthcare people can come here and do an X-ray at your home, nothing like that. And that is where India is heading to in the medical technology segment. But still, with lot of promising sector that we have definitely main challenges here. We have challenges into penetrating into the market, availability of these instruments, adaptability of these instruments. Not many people are saying they are not trusting their own brands. This is made in India, this might not be good. It is same with the research people who, I come from a research organization. I get to hear, if it is coming from Thermo, Kaizen, or some other uh, uh, big names from Europe or US, we know these are set trend and trusted brands, so we trust. A new person coming up with the same machine, we wouldn't trust it. So that is a key challenge in the medical segment as well. So affordability, awareness, accessibility, and availability. The 4 A's are really what do you say, bottleneck for any medical innovation that you are making to enter into the market. It is easier to make, but acceptability by the society is the major crux in this major challenge in this sector. So startup ecosystem, if you talk about, in the last decade, we have more than 1,000 plus incubation centers across India, driven by different ministry, 100 plus by DBT, more than 500 by DST. Startup India also is promoting many schemes for the support of innovation. So we have the ecosystem that in India that has built up. Every city in Odisha itself, we have more than 27 incubation centers focusing on different segments of or different sectors. Ours, where I come from, we focus on life science enterprising, and we support any innovations on medical device, diagnostics, biopharma, food tech, and agri-tech. So similarly, there are Niti Aayog derived innovation centers, which are known as AIC. You will find innovation centers in the name of TBI, Entrepreneurship Cell, incubation centers, Bionest. These are the common names, Makers Lab, plug and play systems. These are the common names that any incubation centers are called as. And they can be from private systems, from government systems, and industry also. And these are the 3 main drivers who are hosting incubation centers in their organizations. And the list that you see in the slide now are the main ones who give funds for your innovation from discovery stage, that is ideation stage, to commercialization stage. So Department of Biotechnology supports biomedical research, and that is the major sector, and they receive maximum application in biomedical sector than any other sector, right? And the same is with DST. DST focuses on different engineering aspects. They focus on healthcare. They focus on uh, uh, environment. They also focus on earth, healthcare, petroleum, pharmaceutics, many other segments. Their school of and the focus area is little wider than DBT. Niti Aayog has had introduced Agni program for igniting ideas. They come up with lot of theme-based hackathons and theme-based proposal calls, where each one of you, if you have ideas to take forward, can approach to these for different funding. Startup India is a recognized body which gives the seed fund for the ideation phase. But for that, you need to start with an entity. Ministry of Electronics, which is known as MITI, runs Samriddhi program, runs Genesis programs, which are used to make prototypes of biomedical healthcare devices or diagnostics. Okay. Millennium Alliance is an fund that focuses on impact, social impact, an innovation that can impact many people. That's where their focus is on. National Innovation Fund is an frugal innovation fund which focuses on rural technologies, either coming from rural farmers and taken into use to the urban or to the society. Like Startup Odisha, in whichever city you are in, you should be having your state-based ecosystem to support the translation of innovative ideas to proof of concept. All right. Similarly, we in Odisha have Startup Odisha, which have different schemes from ideation to product development to need-based assistance, as well as monthly allowance grant to enable any individual who are pursuing innovation on their own. STPI is Software Technology Park of India, which supports biomedical and IoT based software based technologies. Anybody having such ideas and wanting to start an enterprise, STPI opens up a Chunoti challenge. They have a lot of hackathons that comes, and then they also run theme based challenges as well. MSME is more into manufacturing, so they promote manufacturing uh, startups, or if you have a good process that can reduce the time and efficiency for any manufacturing, they would be happy to fund you to establish your industry or a plant. So these are few that I have put up here. But other than these days, Department of Pharmaceuticals, Department of Petroleum, Department of Energy, Department of Earth Sciences, all of this have become active as well to promote innovations in their respective areas. So I would request each one of you to visit the websites and learn more about their schemes and how you can leverage these towards your innovation would be worth pursuing. Now, there are a lot of funding opportunities for student-based, since most of the faculties they are here. And if you are really wanting to start something in your college with your students, I would request you to look at these funding grants. GYTI award, which is going to be open very soon, which is Gandhi and Young Technology Award, which students who are currently working in can put up ideas solving some kind of a problem to even biomedical health problem can be done and a solution can be provided, and they win from 1 lakh to 15 lakh of rupees if selected. Similarly, BIRAC Sitare grant also is for students. You can pass on this information, mentor your students to participate in these kind of funding and grants so that it brings in learning when they apply for those grants. Currently, BIRAC Ignition grant is open. Earlier, they were taking equity, but now they are not taking equity. 50 lakhs grant for any biomedical innovation. So sometimes I keep hearing, our grant is not chosen, our idea is not chosen. I think it is time to look inward and look at the market that we are focusing, a solution that we are bringing in. Is it feasible? Is it scalable? Is it worth pursuing? All these are questions that one has to ask yourself before you're putting any idea towards these platforms, right? Might is from electronics department, and that is entrepreneurship in residence program. Nidhi Prayas, Nidhi EIR are also funds which can be availed right after MS. So I would just want to walk you through a few innovations and how people have taken forward their innovation from here, from India, and then they have reached, created biomedical devices for the for the use of the society as well in local as well as global. Okay, I have divided them into three major categories. Okay? So this is AI digital health in India. Okay? The other category that we often see devices coming in, diagnostics coming in, is a frugal, low resource medical medtech settings. Right? The other is deep science and biomaterial settings. So majorly, what we see here is into these three categories. Now, AI being in forefront, many devices are focusing to create preventive or screening of healthcare systems. So these are a few examples. I will walk you through whom I have seen from their infancy stage to where they have grown. Nirmai is founded by Dr. Geeta Manjunath, and they have been clubbed by an investor. Nidhi Mathur comes from investor side. They have raised investment from SL Ventures. What they are focusing is on a non-invasive, radiation-free breast cancer screening using thermal imaging and machine learning. So she was an academician, and then she left academics and now full-time pursuing her startup in Nirmai. She started with an academic prototype. Then she raised funds from BIG grant. And then she also raised investor funding from Nidhi Asalar Ventures. Then she completed her clinical validation. They have deployed into many hospitals in Bangalore and Hyderabad. And currently, they are looking into global partnership for taking these devices that they have made into the market. So the main challenge while she was developing this screening device was, how do you prove the accuracy versus the conventional method, right? So conventional method, all of us know mammographics are the conventional methods, although they are very painful to the patients, right? Mammography have their own disadvantage. They are huge machines to check, to screen, and they need a lot of big space. They were the problems associated with mammographies, right? So that is where they were coming out with the AI thermal screening for breast cancer, which are more user friendly, patient friendly, and they had to prove its accuracy with the conventional method. When you begin, there will always uh, no trust that this machine will give the same kind of accuracy as that of the mammography. So clinicians were not accepting the studies that Dr. Manjunath was proposing. So it was a lot of hindrance, as I said, acceptance and penetration and support from clinician is equally important to deploy any of these kind of devices into the market. So that is the major hindrance Dr. Manjunath is been facing. And then, of course, the regulatory compliance for AI diagnostics, because the regulation itself is not very clear in Indian context so far. People are still evolving the rules and regulations for these diagnostics. So that is where all of these challenges one are facing when you are bringing in new technology to the market. But what the positive aspect of this innovation is, it is creating a new device with a new technology, and bringing it to the society in India. It'll be a completely cost-effective health innovation, and this can be used in the rural setting as well. Right? So this is the impact that Nirmai was, as a company, wanted to bring in. Of course, any startup's journey is full of challenges as well. It is always thrilling as well as challenges. But I think you figure out a way to overcome all these challenges and bringing the device into the market. If for more information about this device, you can visit their website, nirmai.com. I've put in the slide here. That will give you a complete picture of what they are trying to do in here. This is a very interesting innovation developed by a doctor, Dr. Vishal Rao. He's a Bangalore-based oncologist. So he has developed a voice prosthesis. What this does is it helps the throat cancer patients to speak after their surgery. Usually, when they do surgery, their larynx, the voice box is damaged, and they usually cannot speak. So, what Dr. Vishal has seen in many patients who are suffering with cancer in stage 4, and they were losing their ability to speak because their voice box was not working, it was completely destroyed. So what he thought, if he can make a hole near the neck and deploy this device that you can see the device in here. If you can deploy that device by making a hole in the neck, and if you want to talk, you just close that hole with your finger and speak what you want to speak, and then remove when you're not, remove your finger when you're not speaking. And this gave a new life to many people. And can you imagine the cost of this device? It is only rupees 50. Because the surgeries and then being in the hospital and other things are more expensive. This device he sells it only for rupees 50. That is the change one can bring to the society by creating such kind of innovations, right? So he's a doctor. He's the person who could find where is the difficulty. He came up with the hypothesis. He brought in different engineers together to create a device, then did his clinical studies by himself before launching his product to the market. And he could, since he was a clinician, it was easier for him to deploy because he knew he could believe in his device that he was making, right? So the whole hypothesis for this is the windpipe is connected to the feed pipe, and the air supply from the lungs are able to vibrate into the feed pipe. And here it is that brain trains the food pipe to vibrate again, and that's how the patient is able to speak. You can look at different videos about his innovation, more about his innovation by typing just Dr. Vishal Rao. Uh, and his company's name is Innovation Medical Device. All right. This is another company, Dozy, which is started 5-6 years ago. I don't know the exact years, but they are making a contactless remote patient monitoring system. So this Dozy group is founded by Gaurav and Mudit, and they have created a biosensor mattress measuring the heart rate, the respiratory rates, the movements, and all of these are recorded, and it is transmitted through a cloud-based AI alerts. So how they begin is They identified the problem statement from the ICUs. Okay. And then they developed these biosensors. And for deployment, they contacted the clinicians and wanted to deploy them at ICUs. So first they went to small clinics, and once they were able to validate in small clinics, they did their early pilots in bigger corporate hospitals. Followed by, now they have received government tenders from PAJ, State Health, all of this. Now there's no looking back, they already are into the market right now. So the major challenge that they face is false alert. So when there are too many patients in the room, or there's in the patient bed, somebody else is sitting and things like that, you start to get heterogeneous patient population and false alerts. That was a major problem that they had were facing, right? And integration with the hospital system. It is again the same. All of these medical device people have issues of acceptance and integrations into the hospital systems for leveraging for deployments and doing businesses, right? So they need to rope all clinicians or hospital management people to understand their technology and use their technology. Right. So the learning outcome here was scaling through B2B. They didn't want to go B2C here. They wanted to go do businesses with the hospital so that the revenue and the business plan is big. And also they move forward with government tenders, with public health partnerships. So these are few kinds of devices that you would see have been deployed in last decade by innovators from India. By using the incubation centers as a platform, these people have developed these kind of devices and move forward. Jivatronics is another company based out of Pune who is making hand crank defibrillator for low resource setting. So this does not need any electrical device. So this is innovation of one of its kind, where it uses the dual power. So you see a handle there, you just have to move the handle so that, but the problem that they were trying to solve is unreliable electricity in rural clinics and power outrages. So the hand crank can also defibrillate start to use the defibrillation. And this is used, when somebody has a stroke and he is unconscious, you put these defibrillator in their chest and then give shocks so that they come back, revive back, their heart revives back, right? Sometimes because of there's no electricity in rural setting, this kind of systems cannot be used. Now, cracking to that problem, Jivatronics founder has come up with hand-cracked defibrillators. All right. So now he is supplying even to Africa's to develop less underdeveloped countries as well. So this is a frugal medtech. This comes under a frugal medtech innovation for emergency care and engineering for low resource context. XCO Biosolution um, is a startup who's making rapid homeostasis using a marine polymer chitosan. Right. So what they are trying to make is a dressing that can stop bleed in very rapid, less than 30 seconds to 60 seconds. The bleeding gets stopped when those dressing pads are put into the injury. Right. So they have scaled up this manufacturing unit, and they've also made these products for defense, because that's where it is needed more in quantity, and that is where the requirement is more high. Of course, it is also required in accident places and other things, but they were able to do supply it to the US military grade dressing also. No, they were not able to supply to US grade because it was a challenge to meet their regulatory challenges, right? But they were able to procure the regulatory from the SEO, and then EU regulation they were able to get, because regulatory from FDA and other things are more stringent. So our materials were not able to pass through, but they are selling it currently to the Indian defense as well. So the learning outcome in here is, biomaterials can be used as biomedical materials for stopping the blood within less than 60 seconds. And this comes under class 3 devices. All the medical devices in regulatory categories are divided into 4 classes: class 1, class 2, class 3, and class 4. Class 1 and 2 are high risk devices, and class 3 are little low. More information can be found in axiobio.com. Similarly, I have plenty of cases that we have supported from the Bionest incubation centers to share. This is another startup who is using AI pathology for spear diagnosis, right? Augmented digital microscopy he has used, and AI for blood, urine, semen analysis. So by just checking the smear of the blood, they can give you information without, investing. Sorry, they can give you more information regarding your aspects of the blood, what is the status of your urine, as well as the semen. So the founders, Tathagato and Rohit, has taken up this idea, and they have made a startup, Sig Topal, which right now they have standardized the instrument, and they are already into the market. So the accuracy and data accuracy and sensitivity has reached the conventional methods, but they are trying to inbuild AI modules into diagnostic pathology. One more, I will give a startup that is there associated with us. The company's name is Microbiotics, where he is using AI for testing the gut bacteria to let you know the gut health. So they have formulated a kit where blood can be drawn, it can be tested, and the blood sample can be injected. Based on that, metabolomics is run, and AI is used to understand what are the bacteria missing or what are the bacteria present in your gut, and then a report is given to you about your gut health. So that is one of our startup who has raised 1.5 crores from SL Ventures, is currently incubated with us, and it is a faculty spin-off from NICER. Now moving forward, taking other devices that have come. So these are Manoj and Pratusha, both of them based out of Hyderabad. They have introduced Nemo Care Raksha for neonatals, right? It is affordable, accessible solution to reduce the neonatal death, all right? These are wearable solution, as you see in the picture. The the child has a band around the wrist. It has a lot of sensors and network functions. It gives you a lot of algorithms and big data which can continuously which is helpful for continuous monitoring and preventing of care of these young patients, right? So it is IoT enabled smart wearable watch for the baby's foot. All right, they give parameters like SpO2, heart rate, respiration rate, body temperature, as well as body positions. They have raised $100,000 investment from Venture Health, uh, and they've raised Bill Gates Foundation also have invested in them. They've received funds from Biotech and Chemtech. So with the support from this ecosystem partners, they were able to bring their innovation to the market. This is another interesting device, Soham Innovation Labs Private Limited, who is working again with for newborns. What they are trying to do here is put these hearing device into the neonatals to find out if they have a hearing loss. In most of the times, we don't identify if uh, the the child at birth if he has hearing loss or not. So they have created a device that can predict the hearing loss, or that can find out the hearing loss at the birth in a poor resource setting, right? This is completely Indian made. It is designed for mass screening of neonates in a very resource constrained settings. All right. So it uses brain auditory evoked response technology for the auditory testing. To know more about this innovation, you definitely can follow. I've put their websites here and their contact numbers. So these are the kind of innovations one would look for in, that can make bigger difference and impact to the society. So each one of you must be having a skill set working either in different, sectors of biomedical engineering, right? Some might be expertise in AI, some must be expert in electronics, some must be expert in engineering, and some must be expert in business. I think we just all should come together to work on one common problem and bring new devices to the market, so that we can prevent early death, early losses, and give a better life to each one. Right? I can go on, but I can ask if there is any question. I'll be happy to take. Is there any question? All right.
Participants, if you have any question to ma'am, write it down in the chat box.
Yeah. So I just want to give you a varied varied levels of innovations that have already come to the market, to give you an idea, so that either you can augment the innovation or create a new innovation that can replace the old ones and come with a better treatment plan, better prevention plan. This is another company from UP, Bionic Hope Private Limited, which is creating a low-cost EMG controlled robotic prosthetic hands with a sense of touch for upper limb amputees. All right. It is a 3D printed prosthetic hands with a multi-grip control. I think that was the USP, is the multi-grip control. And it's light-weighted, affordable, and battery-powered innovation that they have done. And it can be used for elbow amputation for the age group of 15 years and above. All right. So these are the innovations that are coming. So there are many prosthetic groups other than Bionic. There are, another dozen of companies who are working on these segments with different kind of material, with flexible materials, with different electronic designs to increase the accuracy and sensitivity of these prosthetics. So that is a different segment, definitely a good segment to work upon and to get as closer and as closer to mimic the human hand or legs. So here you see a wearable wireless limb band for home-based monitoring for Parkinson's disease patients, speech impairment that has caused due to cancer or stroke. It is a simple small device that you see. It can be either placed in the hand or in the limb, okay, which monitors the tremors in the Parkinson patients to regulate the use of the treatment, right? So it has sensors, which can adapt the inertial motion. Unit sensors are implanted into the device. So the first mode is the command that is given by the patient itself, right? They can record it, so it will understand the patient's voice. The second mode, patient can type letters also if they are not able to speak. All right. This is a kind of innovations people are bringing in for Parkinson's neuro degenerative diseases patients. This we just now spoke about. This is the device, that I have already spoke for the voice prosthesis. Arna Medical Bioproduct Private Limited. What they are trying to make is everything in one suitcase available. Okay, they are trying to make silicone prosthesis for breast cancer patients. As you see in the pictures, they are lightweighted silicone prosthetic covers, more giving you a feeling of a natural breast. These can be used after mastectomy, and they are waterproof, and they ease the life of many of the breast cancer patients. So Arana Biomed Medical Product Private Limited is coming up with this device, which is giving ferment, which the material that we use is very user friendly, very closer to human real breasts. So these are much lighter and more normal than the imported prosthetists that are available. Now if you look at, we have so many diseases that we can work upon, right? So each disease has so many treatments, so many solutions that one has to offer. And we can definitely incrementally add on innovations to the existing solutions, or come up with the completely disruptive innovations to give a better feeling to the patients and make their life even more comfortable. So, these, when you think of where do you get an idea, most often when I go to colleges, when I go to faculties, or when I go to researchers, he says, we have a skill set, we don't know how to use it. And how do you ideate? From where do we begin? All right. So there's always businesses lies in three things. Somebody's laziness can be somebody's business. Somebody's pain can be somebody's business. Somebody's happiness can be somebody's business. So if you are able to tackle any of these three, you know where you can add. So it can be in any segment that you are working. So this is another company who is making a low-cost, multi-powered, multifunctional neolateral breathing system support device. As you see, this is so compact that Incel is a company who has introduced the device. Device is known as SANS. It's a portable, easy to use, robust neonatal CPAP device for short-term breathing for infants, which are there in neonatal ICU settings, as well as there are less resource settings for neonatal people. There, these kind of a device can be used. So these people also have used the funding from BIRAC. They've used the funding from Nidhi Prayas. They also raised funding to bring this innovation to the market. So B4 Labs Private Limited is introducing a video laryngoscope for use in emergency room and operation theaters and surgeries. They are easily intubated with a video, as you see in the picture, right? The channeled disposable blade with an attached camera goes into the laryngoscope, and the picture that is seen outside in a bigger scale. So it is a smooth self-guided uh, device, the laryngoscope, right? Nowadays, you hear most of them talking about remote surgeries, robotic surgeries that can happen. All of these are happening, is possible because of, um, you know, the smooth, self-guided cameras that can be implanted with the instruments and that can go inside our body to take the real-time pictures of the body. So people can really work on, the day will not be far away where doctors would be doing surgeries remotely. Although, I hear people have started, but I have not myself experienced or in my near family, but people have started doing remote surgeries because of the technology advancement, bringing in AI, bringing in machine learning, embedding small cameras available in just like, miniature size that can go, and materials that can go inside our bodies and not being toxic. There are a lot of innovations have happened in those areas which are enabling these kind of devices to come to the market. So this is Rajesh Laboratory, who has come up with a novel pocket medical scanner, which is a non-invasive medical diagnostic device based on sound analysis to diagnose and screen diseases for underdeveloped and developed countries, right? So this is a potential to diagnose, screen respiratory devices and cardiovascular devices. So any of you, if you're working on TB, I think government's focus is on TB Mukt Bharat. So any innovation you're making on those lines will be taken forward and you would be more appropriate for such initiatives taken by the government. So please reach out to either the incubation centers or to the people who are promoting the TB Mukt Bharat or pneumonia, uh, those grant officers or those officers, you need to reach out so that they know that you're working on these kind of ideas to take it to the next stage. Here you see an innovative solution for prediction and management of epilepsy. Right? You see smart gloves, which is capable of sensing electrical signal from our body, keeping a track of our health. All right. And then it'll let you know when you're going to sleep, how you're doing your daily routine and other things. This gloves is named as TJ. This is designed for early detection and management of epilepsies. Now, these gloves have several sensors and which can extract information from the palm of your hand and continuously, and then they are analyzed and in a cloud environment under certain intervals, and the information is passed on. See, the innovation of making sensors smaller and smaller in size. People working on those electronic departments, working on hardwares and all, are doing an incredible job in making sensors more effective, more accurate, and in smaller in size, and also compatible for many situations, right? So this is a device that is used for epilepsy. This is another technology from Flex Motive Technology Private Limited. It is a flexure kinetic shaped crutches for enhanced mobility and reduced energy consumptions. This is mimicking the human feet, and it can be stable in uneven surfaces as well. All right. So this was designed to have a rubber gripping so that you can hold it tight, and it prevents the slipping in the the smooth surfaces, wet surfaces, snow, sand, and mud. These have been tested. So this is done by flexomotive technology. So there are plenty of such devices that are innovated, made in India by the Indians, available for Indians, right? So we can use our skill set and do multiple other innovations by collaborating, by uh, promoting, encouraging, whoever are there in whatever stage. If they try to contribute to the society, they should be enablers, should contribute in enabling the innovators to come up with new innovations. People sitting in the government should help it with easing out the policy and to let the innovation come forward. People, investors having money should promote these kind of innovations. So I think if every stakeholder in this ecosystems come together, we will have definitely more meaningful innovations in the medical technology. With this, I would stop my presentation, and I'm open for any questions if you might have for me. My phone number and my email address is there in the website on the slide at the corner bottom. If anybody wants to reach out, they can reach out to me personally as well, but we are open for questions. Is there any questions, ma'am? Is there any questions from the participants?
Until now, there is no question.
All right. Yes, sir.
Participant, I hope there is no worry. If you have any worry, you write it down immediately on the chat box. Over to Ranjit sir. Yes, sir. And one question has come in the chat box. You can see in the chat box, and I will read it out.
I'm just checking the chat box. Can epilepsy be detected with MRI imaging? I'm actually not sure about epilepsy detecting by MRI imaging. That's more technical of a question. But I'm sure there would be changes in the brain signal when this kind of incidents happen. I think that is why CCDs that they put to check if there is any disturbance in the brain network and signals. If person collapses collapses and is unconscious, and I'm sure they should be brain CT scans should be able to tell us the difference. Um, there should be some different recording than the normal recording of the brain, and the answer would be yes. Brain CT scans are already there, right?
Is there any query? Is there any other query? No. Over to Ranjit, sir.
Thank you very much, Madam Dr. Nivedita Jena, for your insightful and inspiring session on case studies, startups, entrepreneurship, and innovations in medtech. The discussion on startup ecosystems, real-world challenges, and innovation pathways has truly broadened our understanding of the medtech tech landscape. We are grateful for the practical perspectives and motivations you shared with all our participants. Thank you, madam.
Thank you so much, ma'am, for your insightful session.
Thank you.
Thank you.
Thank you.
Good night, ma'am.
Thank you. Good night. Can I log off?
Yes, ma'am, you can log on.
Okay. Thank you so much. There's some question they're saying multimodal imaging for epilepsy detection is possible. I think it is possible. You know, a lot of people who have started working on multimodal imaging, I think, for epilepsy. You need to just check more detail and how they can be done. Because if I recall, they are PET imagings, they are MRI, ECGs, they are combining multiple imaging systems to create new innovations and make the what do you say, MRI scans more accurate and more informative to bring the advanced version of MRI system. I think that is possible, sir. All right. Thank you so much for having me here. All the best to all the participants. Thank you.
Now moving on to our second session of day 5, we will be focusing on a critical and rapidly growing area, that is big data in healthcare. We are honored to have with us Dr. Ramakanta Mahanti, who brings deep expertise in data analytics, healthcare informatics, and research-driven innovation. To formally introduce our distinguished speaker, I now invite Mrs. Pratiti Mishra to present the speaker introduction. Over to Pratiti, ma'am.
Thank you, ma'am. Good evening, everyone. It is my immense pleasure to introduce our esteemed speaker for the second session, Dr. Ramakant Mahanti. Dr. Ramakant Mahanti holds a PhD in machine learning and soft computing from Barampur University in Odisha, as well as MTech in software engineering from NIT Allahabad and BTech from Calcutta University. He has published 89 papers with 24 in Redford International Journals, 50 in Redford International Conferences, and 5 in invited book chapters. His research has been published in journals such as Applied Subcomputing, Soft Computing, Decision Support System, Expert System with Applications, Engineering Applications of Artificial Intelligence, IJCIA, IJAEC, IJIDS, and IJDATS. He has served as a PhD supervisor at JNTUH in Hyderabad, Barampur University in Odisha, University of Mumbai, Gitam University, His research interests include fuzzy computing, neurocomputing, soft computing, evolutionary computing, data mining, text mining, wave mining, big data, privacy preserving, data mining, fraud analytics, sentiment analysis, social media analytics, big data analytics, and churn prediction in banks and firms. He is on the editorial boards of IEEE Transactions on Software Engineering, IEEE Transactions on Knowledge and Data Engineering, and Elsevier journals such as Information Sciences, Journal of System and Software Knowledge-Based Systems, International Journal of Software Engineering and Application, and Journal of Engineering and Computer Innovations. He is also associated with IEEE conferences like CSMT, BICA, SCN-CCO, FAN-CCO, CICN, and Springer's conferences such as FAN-CCO, SCN-CCO, IC3T, ICCII. He has been a member of the International Technical Committee for conferences like WICT, CSMT, BICA, NWSP, ICACA, ICAKE, and others. In India, he has chaired a number of IEEE and Springer international conferences. He is the principal investigator and co-principal investigator for 3 government-sponsored projects, including SCRB Core, DST, and AICT, and took national patents in his credit. So such a rich profile. Please join me in warmly welcoming Dr. Ramakant Mohanty sir for his expert session on the topic Big Data in Healthcare, Tools, Ethics and Insight. Over to you, sir.
ಇದು ಆನ್ नहीं गला
Sir, you can say it.
yeah so let me share your screen. Yeah. So my PPT I will share now. Yeah. Yeah, good evening to all. Yeah, myself is Dr. Ramakant Mohanty. Presently I am working as a professor and IT chairperson in ASBM University, Bhubaneswar. Is it audible now?
Yes, sir.
Is it audible?
Yes.
Yeah.
Yes, sir.
Fine. So my talk today is Big Data in Healthcare, Tools, Ethics and Insights. And my subtopic will be transferring healthcare through data-driven decisions. And this particular presentation will be shared by 2 persons. Myself, I'll talk about the introduction about Big Data, and case studies will take care of by Aishwarya Priyadarshini. Regarding overview, today we'll talk about introduction about Big Data, different challenges, research gaps, problem statements and objectives of Big Data analytics. Here we'll talk about case studies that propose technique 1, then propose technique 2. Then we'll discuss about how to take a data set of large data sets exactly of size, very high size. Then how we'll do segmentation. Then how we'll apply different type of machine learning algorithms to find out the infrastructures in. Basically, we'll take a case study of cancer, lung cancer, and we'll show you step by step and the process involved for doing all the things. And that part will take care of Surya Priyadarshini. And she will also discuss about how to visualize all the information related to big data. Then discussions on what kind of results you can expect by taking a large data set, and future directions also will discuss by her. And I'll also take care of conclusion and references, etc. Now see, introduction to big data, particularly in healthcare. I hope everybody knows about what is a big data. And if we talk about big data, simple case I'll just take. Suppose you are going for a Gmail, and Gmail is having a capacity of, let us say, some 10 to 15 megabytes. If you wants to send a data of 100 megabytes, that's become a big data for the Gmail. But exactly if we say big data definitions, many people might have told, I just speak only few words. Big data are of large size, and that size differs from problem to problem and people to people. And here we'll discuss more detail about healthcare, because my topic is healthcare, big data in healthcare. And we'll talk about data size of not less than hundreds of terabytes. If we'll speak frankly, what is a big data? That size may vary, as I have told, problem to problem. It may not be gigabytes. It is above exabytes nowadays. And just you can take a simple case, the mobile data all over the globe. And as per the statistics from 2020, it was around 4,000 exabytes, not hexabytes, exabytes. And if we talk about 2025, it crossed more than 5 lakh exabytes. And if we talk about more details about in healthcare, the MRI scan reports, it becomes now in exabytes only if we talk about global scenario. And patients all over the globe, if they are going for MRI scan, and if the report is available in some large sources or storage devices, and it can be a huge amount. Another area of this big data is bank transactions, fraudulent bank transactions, and how to find out that huge amount of data is a big matter in his life. Therefore, the topic is excellent, and this is very fast growing area where many people have started working and finding a very excellent results. And that gives a solution to the doctors before prescribe a medicine. They can able to access those data, those patients, and able to prescribe the correct medicine for a particular patient. Next comes why it is big data. Same thing I discussed already. Every second, vast amount of digital informations are available or generated from our daily lives, from financial transaction to social interactions. Therefore, big data enables data-driven insights, real-time analytics, and evidence-based decisions across the industries. And if we show what could be the different types of big data, already I think some people might already speak about this. This is volume. Volume starts from, as I have told you, it is problem to problem, person to person, department to department, country to country. So managing large volume of data, that's why you should say it's a large volume. Nobody will say that big data will be this much size. No, it can be anything. Next point, I have told velocity, then variety, veracity, value, and one more is there that is called variability. That also I have not added. There are 6 P's in big data nowadays. And this variability is related to data inconsistencies and the data flow and how to storage those huge amount of data is a condition of is called variability. Another is big data. Then I talk about why healthcare needs big data. First of all, we have to talk about EHR with analytics engine. Then if data is having that much storage, where will store? What kind of tools are required to analyze them? How to analyze? Next is called, as you know, that AI and machine learning, I will talk about this matter of, you see, AI is the umbrella of all the things. AI has come, neural network has come, machine learning, deep learning, generative AI. Now so many things are coming day to day to solve complex problems of huge data size. And big data, then next starts big data platforms and where you will analyze it and what is the cloud warehouses mean storage, where will you be able to store this amount of data. Then comes different type of sensors will be used in different to acquire or to store to the data, that is called, we'll use IoT devices. And somewhat you know that all over the globe there are so many countries and there have so many languages. We need there are also to convert their language into a human or computer understandable languages. For that we need natural language processing. Then genomics and bioinformatics platforms that are also bioinformatics analysis of bioinformations, particularly healthcare systems like different type of diseases. And that should be a separate domain where we can able to store and not only to store, to understand easily and to make them understand others, we need data visualization and BI tools. Especially Tableau and BI tools both are used for this purpose. Next comes what constitutes the big data in healthcare. First is EHRS. What kind of data will be? See, data may be structured, unstructured, semi-structured, but we need to convert all EHR reports into structured data. And examples of those EHR reports are patient demographics, diagnosis, lab results, and medications. Then comes medical imaging. Imaging always will be unstructured, and we should have a separate tools to make it structured data. Then only we can able to analyze by employing machine learning and AI techniques, deep learning, ChatGPT, etc., etc. And these are can be X-ray, MRI scans, CT scans, and histopathology slides. Next comes genomics and omics data. These are very high dimensional in the order of maybe as we have worked 2-dimensional or maybe 3-dimensional. It is having many dimensions. And for kind of like examples are DNA sequences, gene expressions, and proteomics, examples of genomics and omics data. And wearables and IoT devices also required. See, as you know that many people have started using IoT devices in agriculture, used in different domains. But nowadays, IoT devices are extensively used in medical industry to collect the informations from time that is real-time streaming of data. That is what devices will be placed in heart rates, glucose levels, and activity tracking. All the things are required to store the data by using IoT devices. Next is called clinical trials and research databases. These data are semi-structured in nature. Semi-structured is easily you can convert it into structured data. And these data can be of trial outcomes, drug efficacy, and adverse event data are the examples of clinical trials and research databases. Similarly, most important is social media and patient feedback. That will be in the form of textual or sentimental. Here also sentiment analysis also required to find out what is the view of patients, and what is the view of social media. Based on that, patient opinions, public health trends are examples of big data in healthcare. And last is your administrative and insurance data. These data will be structured, and like billing records, claims data, resource utilizations are the examples of all healthcare systems. Am I audible? Am I audible or not?
Yes, sir, you're audible.
Yeah, fine. Then because time to time I need to ask.
Yes.
Next comes, what is the basic architecture? See, many people, they have shown many architectures, might have shown, but this is what I feel the minimum architectures. Data sources. Data sources, already I have told many things about data sources. And injection. What do you mean injection here? Data sources will be many, and each data should be stored separately for a particular purpose. And we need to divide and keep in a safe place. And as you know, that big data, mostly people know that we will have to work in either using supercomputers or maybe the cloud. Therefore, storing and separating data of different sources and utilizing for a particular purpose is a matter of concern. Next is called stream processing. Here, again, data size, as you know that data size will be of minimum terabytes of data. Then we need to process the data. Processing it by normal computers or by laptop is not possible. Therefore, either you need a lab where your supercomputers are connected, or you have to take a help of a cloud to stream the data of different kinds. And finally, you need to store for a particular purpose. After storing, you should know how to analyze and visualize the data before going for analytics. And if you understand all these 3 to 4 parameters, then further is the job of a AI to analyze and give a particular decision based on a particular problems. Tools and techniques. I think there are 20 tools are available in the market, but the most used one, just I will just tell you 20 tools are there. Let us start with one. Most important tools are those you are using nowadays. Is a Hadoop Spark. Then Talent is given by NIFI. And others are Hadoop, you know, as it is a basic level, that is called SDFC and MapReduce techniques. NoSQL, that is called MongoDB. This is for storing, and Cassandra is a database. MongoDB already there. Cassandra is also there. This is the where you can able to store large amount of data. And for any queries, normal SQL queries may not work. Either you have to go for NoSQL or MongoDB database, or another is called PostgreSQL. PostgreSQL nowadays they are using for any queries for large databases. Normal SQL query may not work. In addition to this, many other tools are there: Hive, Pig, Flink, Kafka, then HBase, Resto, Elasticsearch, Splink, and like if you want to talk for analytics and visualization, Tableau and Power BI, these are the two important things that are there, but Tableau is more comfortable compared to, I hope, Power BI. Then Talent, already I spoke, is already there, data integration. Apache NIFI also already there, NIFI is made by the Apache. Then we'll have RapidMiner. RapidMiner, these are called data, what is called machine learning tools. RapidMiner is tool used for data analytics or prediction, etc. And there you will have like supercomputer machine, many other things. NIME means you will have like all decision trees like CART, Relate, Mars, and random forest. This is another software It is student version is available as well as paid versions also available. You can use out of anything. Then another is called data robots. Nowadays, this is a licensed one. It is not free. Maybe it is given student versions will be available, but you will find some other sources. So these are called tools and technologies used for processing, analyzing, storing, and for analysis purpose, as well as for analytics and visualization purposes. Most important point here I have talked that C, C++, Java is no more works in big data. Either you have to use Python or R, and another new language has come into market now is called Julia. Though it is not that much uh, used in real time, but R programming and Python is used for analyzing this data. Java, C++, C is no more for this large amount of data. So what are the applications are there already? Many people might have spoke. Big data applications in healthcare, particularly in my area. And we can talk about disease prediction and outbreak control, clinical research and trials, drug discovery and development, precision medicine to be given by the doctor, you have to say how, whatnot. Then population health management, clinical decision support, and personalized medicines, and finally, predictive analytics used for medicine purpose. So these are the basic things. Here, many case studies you can take. Data available in the Kaggle as well as UCI repositories. Main thing is that how to download and where to download. That's the big issue. And after that, how to segment the huge amount of terabytes of data. That is another big concern. Without the help of clouds, it is impossible. Or you can take, suppose in big organizations like IIT, they have supercomputers. You can download because they are having a capacity of exabytes of data you can store. Accessing up to that level, a normal faculty or maybe a student is impracticable. Let us see, I just spoke about what are the data sets are available of size above terabytes. COVID-19 and data analytics is available almost, I think, in the repository of KD Nuggets, might be knowing KD Nuggets place where data size is around 8 to 10 terabytes and up to 50 terabytes also available, but accessing and downloading with the normal system is not possible. Next comes how to do predictive analytics in big data. Here just I'll give you simple steps. Main idea behind is this segmentation. Because if you want to work in a particular system, first you have to download by any sources, any methods, or you can take those access of cloud by paying some amount and ask the tools there to segment the data into small parts, not more than, as let us say, 10 to 20 GB. 10 to 20 GB in a normal system is not possible to, even also to process. It takes a lot of time. Or below 1 GB, segmenting data. So let us say you are having 1 exabyte of data and segment into less than 1 GB data. Then though you cannot get a very good accuracy, but you can able to show, and doing by, let us say, deep learning techniques, you can give some kind of results which may not be 100% accurate, but approximately, because you cannot process this data, huge amount of data with a small laptop or something like this. So another is called diabetic radiopathy detection systems. This is also available data in same areas, or maybe another is called TCG. TCGI, my daughter will say, Aishwarya will say about where data is available, and she worked for almost 2 to 3 years and still working in the field. And brain tumor classification using MRI data. and histopathological cancer subtyping, all are called different types of data related to big data in health. And I hope this part of program will take care of by Aishwarya Priyadarshini. Aishwarya, are you available?
Yes,
Aishwarya?
hello, sir. Yeah,
Ah,
I am available. Yeah.
So she will take care of this part. Last part, I'll talk about conclusion and what we should do, because as a faculty and where you can start, what are things you have to do so that you can able to do a small work. Let us say those who are doing PhD in this field, they can have a clear ideas. So Aishwarya, hand over to you. You can go ahead.
Yes, sir. Only thing is, can you keep presenting? I'll just ask you to move on to next slide, so it'll be easier for me that way.
No, she will do it. She's her end also. I hope she can do it. Everything. Okay.
No, you're already presenting, right? So,
Yes,
I will
yes,
stop.
yes, visible. And madam, a warm welcome. Achara madam, a warm welcome to our FDP. And you can start your session. We are inside. We are excited to listen from you.
Yes.
thank you so much for the warm welcome, ma'am. Give me a second, please.
No,
Sir, can you please have it on mute? You're speaking. I can hear you on the call.
I'll fine.
Please mute yourself. Thank you. I hope I'm audible.
Yes, ma'am, you are audible.
Yeah. Thank you so much. Thank you for your patience, everybody. And thank you, sir, for introducing the subject big data in healthcare. Good evening, everyone present here. I'm Aishwarya Priyadarshini. I'm currently working as an R&D engineer at Samsung Research, and my presentation today is based on the research work carried out during my master's at Indian Institute of Technology, Indore. To maintain my bandwidth, I'll just turn off the video while I present, right? So thank you for your understanding on this. Uh, can so before I start, I'm truly delighted to be here today and to have the opportunity to present my research work as a case study under the theme of big data in healthcare. My talk today revolves around the fascinating and crucial domain of, fascinating subject, digital histopathology, and how artificial intelligence, AI, generative AI can assist in automated classification in the field of diagnostic histopathology. Before I move into the technical details, let me first give a brief overview of what this case study is about and what we'll be exploring together today. So, histopathology, in the simplest terms, refers to the microscopic examination of tissue samples to study the manifestations of a disease. All right. It's a cornerstone of cancer diagnosis, where a pathologist examines tissue slides under a microscope to identify whether the tissue is benign, malignant, or which type of cancer it corresponds to. Now you might be thinking, what exactly am I referring to? Basically, histopathology is nothing but, analyzing your cells in the body or the cells from a tumor region to understand whether it actually can be cancerous, it's deadly or not, if it's benign, does it need any treatment? So these are some of the procedures that you can actually do when you study the cell structure. And this is something that is carried out by the pathologists. And the tissue, the thing that I'm referring to tissue is basically it's an area cut out from the tumorous region and viewed under a microscope. So that is what I'm referring to as tissue. Now, in recent years, with the advancement of digital pathology scanners, these glass slides. So, what exactly is done is the tissue that you have sectioned out from the tumorous region, it's put on a glass slide, stained, and viewed under a microscope. Just like you might have done some basic experiments when you were all kids, maybe around eighth or ninth standard. You used to look at plant cells. You used to section out plant cells, put them on a microscope, put them on a glass slide, stain it, and view under a microscope. It's the same process. Now, however, it can be digitized, and we call them as whole slide images. So, when it is a microscopic image, it's absolutely a very high resolution image, and it contains gigapixels worth of size. So, one image can be 2 GB to 3 GB, and sometimes even 10 GB. It's not your everyday cat or dog image that you see, or the selfie that you take. At max it could be 50 MB, 60 MB, with the best of resolutions that you can capture an image. But the image that I am talking about, one image of it, a microscopic image, is at least 2 GB to 3 GB, and can go up to 10 GB as well. So these are extremely high resolution images, often several gigapixels in size, and they capture every microscopic detail of the tissue specimen. And this transition from traditional to digital histopathology has opened the door to computational analysis and AI driven diagnostics, making it a prime example for big data in healthcare. However, processing such data is far from trivial. As I said, it's not your everyday image that you take in. So, as most of you might already be aware and working with AI, the general size that a machine or a pre-trained model that we'll be talking about later can process is 224 cross 224 pixels, or maybe 256 cross 256 pixels. But this, I will show you that these are around 100,000 cross 100,000 pixels and definitely not machine trainable. These images are massive, complex, heterogeneous, contains million of cell information with subtle variations in color, structure, density, and each whole slide can be tens of gigabytes in size.
Sorry for interruption. Actually, your PPT is not moving. You are in the first
No, no,
slide.
no. That's right. It's just an introduction of why we have taken up this topic. The next slide would be about, the in-depth details about it. So yeah, this slide is correct.
Then it's fine, ma'am.
Yeah, yeah, yeah. It's fine. We'll look into the details. I'm just explaining why, histopathology is actually big data in healthcare. So now, so this makes it a perfect example of why we call it a big data in medical imaging, because it has high volume, high density, and high variability. So in my presentation today, I'll be discussing how we handle these challenges using AI. We'll start by looking at the motivation behind focusing on the problem statement digital histopathology. And so to begin with, let's first understand why lung cancer. So there are multiple types of cancer out there. And as you can see, we have selected lung cancer as the basis of our study. So let's first understand why we are going for lung cancer. So lung cancer continues to be one of the most prevalent and deadly forms of cancer worldwide. According to recent global cancer statistics, it remains the leading cause of cancer related mortality between men and women both. So, what makes it particularly challenging is not just its high incidence, but also the fact that early detection can save a lot of lives. Broadly, as you can see from the pie chart, lung cancer is categorized into two major types: small cell lung cancer and non small cell lung cancer. And within that, lung adenocarcinoma and lung squamous cell carcinoma make up the majority of lung cancer cases. And um, as you can see from the image, what I'm trying to say is we have historically always tried to, classify between whether a tumor is cancerous or benign, mostly benign or malignant. But when it comes to classifying between the subtypes of lung cancer, it is a very new topic to a lot of people. Any type of cancer has many types of subtypes in it, and it is a very rarely discovered topic. Can you please go back to the last slide, please? Yes, thank you. So, as you can see from the pie chart, only 65% of the entire lung cancer cases, which is the majority of the cases, are lung adenocarcinoma and squamous cell carcinoma. And why are we exactly looking into the subtypes is because, the treatment procedures that are used for lung cancer is not the same for every subtype. They actually have different treatment strategies for different subtypes. And if you say a person has lung cancer, there is not one target treatment that is done. They look into the subtypes, they understand what is going on, and then they actually decide for the treatment. So, our study is going to be to classify between the subtypes of histopathological lung cancer. Next slide, please. Okay. So now, so we just saw what is subtype classification and why is it so important. So traditionally, we always talk about cancer diagnostics, which is mainly about a tumor being benign or malignant. But in our case, we'll be looking forward and trying to understand how we are going to differentiate between the two subtypes of lung cancer, lung adenocarcinoma and lung squamous cell carcinoma. They differ significantly in their histological patterns, genetic mutations, and most importantly, in their response to targeted therapies. For example, certain drugs are effective for adenocarcinoma but completely ineffective for squamous cell carcinoma. So, accurate subtype classification directly impacts treatment planning, drug selection, and ultimately patient survival outcomes. That's why our study focuses not just on identifying lung cancer, but also differentiating between the histopathological subtypes. Now, when we think of diagnosing lung cancer, there are several imaging modalities that immediately come to mind, right? For example, MRI scans, CT scans, PET scans, chest x rays, and everybody, almost all of us have heard about this, right? But the thing is, these are routinely used and they are generally to detect whether there is a tumor or not. It doesn't say anything about the subtype. I mean, it can tell you, okay, the person has lung cancer, but then what type of lung cancer is it going to be? This is not something that these images can give you information about. So they can give you the location, they can give you whether yes, a tumor is present or not. So for us to actually differentiate between subtypes, go in detail, we need to understand the cell morphology. And that is not something that can be given by these images. So, what we exactly use is biopsy based diagnosis. Basically, as I said, it's a tissue sectioned out from the tumor region of the cancerous area and put on a glass slide, stained and viewed under a microscope. So we also call it as biopsy. In the next part of my presentation, we'll look at how biopsy samples are taken and how they're digitized into whole slide images and how we are going to use AI to actually understand and automate the classification of these images. So next slide, please. Yeah, okay. Motivation and challenges, fine. So in recent years, there has been a growing demand for diagnostic support tools that can assist pathologists in handling the ever increasing volume and complexity of medical data. So this is why computer assisted diagnostics comes into play. Our motivation for creating a computer assisted diagnostic system for digitized histopathology imagery is because this image is huge. Okay, the complexity and the scale of histopathological images is not comparable to any other type of images that you see in general, or even the medical data uses in general. Like the X-ray images are really, really small compared to what images that we will be using. So storing, processing, and analyzing such massive images is computationally expensive and time consuming. And beyond the technical challenges, there's also human challenge. For example, there could be inter-observer variability. That is, when two pathologists interpret the same region of tissue differently. So when you actually go into the minute details, morphological cues of a whole slide image, you're going to see that, there are so many differences that even two pathologists looking at the same region in the image could be confused and arrive at different decisions. So, these challenges, the complexity of data, computational cost, and the need for consistent diagnosis, collectively forms the motivation of our research. So, next slide, please. Okay, so I would also like to discuss, what kind of work has been carried out in this area. Over the past decade, there has been remarkable progress in applying AI to medical imaging, big data, healthcare, and all of this. We have been hearing a lot about automating AI systems, introducing AI into medical field so that it can help doctors have a proper prognosis before they actually make a decision, diagnostic decision. So there is a lot of progress. So coming to the research part of it, I will give you one or two examples of how people and researchers have worked around this. To start with, one of the early studies by you et al. Also not very old. It's in 2016 or two, and so on. Can you go back to the last slide, please? Please don't change the slide if I don't say so. Another slide back, please. Yes, thank you. So we'll be discussing one or two of the research works that have been carried out in the past, and yet, as you can see, the first work is in 2016. So this subtype classification has just arrived in the market, or people have started working on it just 10 years back, not even entirely 10 years back. So this is a very new problem in the research field. And to start with, you et al., as you can see, combined medical conventional image processing techniques with machine learning classifiers. So basically, they use some handcrafted features like you do in your conventional machine learning, where you already have features defined, and then you train your model and try for prediction. They did the same thing and they got around 75% accuracy, which I would say was a promising result at that time. But considering the complexity of cellular features and microscopic variations, this is not enough and not considered a good result in a medical field. It has to be at least 95% and above to be considered as a good result and to be actually deployed in the medical field. And similarly, you can see that, Chen et al., they use the Cancer Genome Atlas dataset, which I'll be talking about in a bit. It's a very good data set which is publicly available. All of you also can go ahead and look at it when you have time. So they use this data set. This contains subtypes of lung cancer, and there are images, genomic data, a lot more, bioinformatics profile, complete informatics profile of a patient. So you can go ahead and look at it. So what they did is they used this data to train, convolutional neural networks. Many of you would have already heard of it, CNNs or deep learning, and they actually use the entire slide data to, train their models, which is basically, uh, training on the entire image 10 GB of data at a time, which is actually not really feasible. Even if you look at your current systems, it's not feasible to train on 10 GB of data at a time. And if you have 1,000 such images, you have like 10,000 GB of data you're training at a time, which is not really feasible. So even if they get a good accuracy of say 0.83, it's actually not really, useful in our case. So moving forward, there are other people who used other emerging techniques in the field of deep learning, for example, ResNet, vision transformers, and so on. You can go ahead and search this up and you will find so much of research in this area. In the recent years, just 2 or 3 years, you'll find a lot of data and you can actually go through it. So after carefully considering the literature, we identified some research gaps. So we'll be discussing those research gaps and how we addressed it. So can you move on to the next slide? Okay. So as you can see, I have outlined some of the research gaps here. So first thing is, as you can see, we have our first image here. This is how a glass slide looks like. Okay, and the staining of the tissue taken from the tumorous region and stained and viewed under a microscope. So this is what it would look from a very high perspective. This is just a small image. But when you enlarge it, as you can see, very small regions will open up to show the subtype 1, subtype 2, and regions like this, right? So this is a very big image. But the issue with this is we have limited annotated data availability. So the data that we have about this image or the classification label, whether this is lung adenocarcinoma or lung squamous cell carcinoma, is only at the slide level. So we can see the picture and say it is, lung adenocarcinoma or squamous cell carcinoma. But now, as I already discussed, the differences between the subtypes of lung cancer is only understandable when you look at the cell structure, right? And from this image, from the top, you cannot really view anything about the cell structure. So even if you have the slide view and slide classification label, it's absolutely useless. You need to understand these small labels, the small squares that you see. You need to understand what, cancer subtype do these subtypes belong to. Only then you'll be able to understand various parts in the entire slide and tell whether the entire slide is having lung adenocarcinoma, lung squamous cell carcinoma. So this is a huge limitation in our problem statement. The next is suboptimal diagnostic accuracy. So we have already discussed this. Since this is a very big image, two people looking at the same square may be confused as to exactly what cancer subtype it belongs to. Can we go back to the last slide, please? And there's this high hardware requirement and computational overhead. So again, this is about the same thing. You cannot train on tens of thousands of GB of data at a time. If you look at your own computer or the PC you're sitting on, it has RAM of maybe 8 GB, 16 GB, or 32 GB, and at max a storage of 256 GB or 516 and plus some SSD or maximum 1 TB in most cases. But the entire data set is just more than 1 TB. You cannot really even have the data set on your system and process it. So obviously high hardware requirement and computational overhead. So these are some of the research gaps that we will be addressing in our research work. Next slide, please. Okay, so we have already understood what we are going to do. I'll summarize it for you in a bit, in 2 lines. Basically, we want to accurately classify the lung cancer histological subtypes in lung cancer, and this remains a challenging task because we do not have the region-wise annotation, which we also call as pixel-wise annotation. And there could be similarities between cellular morphology. You have to have a careful examination to understand this. Highly computationally complex, requires large storage areas, and it's also subject to inter-observer variability. When the pathologists can have confusion about the regions that they are diagnosing, we are just human beings training some pre-trained models, right? So we are going to be addressing the research gaps that we saw in the last slide, and this is our problem statement. Next slide, please. Okay. So here I'll be discussing one of my research works that I had proposed. You can go and look it up. You will also find it on the internet. It's a published paper. So in this, basically, we have proposed a global context-aware network for lung cancer classification in whole slide images by the name eGlocoNet, which is a weakly supervised, advanced attention augmented deep convolutional neural network to efficiently classify lung cancer histopathological subtypes. So here we'll not only see how we process these histopathological whole slide images, but we'll also see, what kind of deep learning we are using. Since this is a case study, I would take you through all the steps involved in this pipeline or framework where we entirely pre-process the given images, we train our model, we perform classification, and we see the results. So we are going to look at the entire pipeline. And you might have some questions related to the AI or deep learning framework, which I am using, or maybe in the pre-processing steps. I'll take those questions at the end of the presentation. So let's broadly look at the steps involved in the proposed approach. So collection of raw digitized histopathological whole slide images, foreground information or extraction, tissue segmentation. So as Mohanty sir already discussed, segmentation is going to be a very important part of processing big data in healthcare or any big data that you have, because it's absolutely not directly machine trainable, or you cannot, directly use it in any form. So segmentation is the most important part in pre-processing big data. Any kind of data you might take, textual data, or numeric data, anything you take, you need to convert it into some meaningful segments which can be understood, and from where the feature extraction or the features extracted are actually meaningful. We perform something known as sampling and stain normalization, morphology prevention, preserving augmentation pipeline. We train the model, and then we perform classification. Let's look into the details of it. Next slide, please. Okay. So as I said, we have used the Cancer Genome Atlas TCGA dataset. Okay, this is a publicly available dataset. If you guys want to experiment, in your leisure, or you want to look at how the images are, they actually have an image viewer tool as well, where you can magnify the given whole slide image 40 times and look at the cellular structure yourself. So I have provided the link in the description. You can go ahead and look at this data set, explore various things. This is a US based data set. And we have around 1,053 samples in our case study, out of which 541 samples are of lung adenocarcinoma subtype and 512 samples are of lung squamous cell carcinoma subtype. And they are captured at 40x magnification. And we have only slide level classification labels for these images. Next slide, please. Can we have the next slide, please? Yeah, thank you. So we'll now dive into the technical aspects of it. So I'm discussing the dataset partitioning specifications. So we have a total of 1,053 images, as I said, in our training, in our dataset. So we have done a five-fold cross validation. We have carried out a five-fold cross validation. And as you can see, we have maintained the train test split ratio in the ratio 9:1. And from the train test, we have again maintained a ratio of 8:2 for training and validation. I have prepared this table where you can see the distribution of number of samples for train, test, and validation. So normally, in a normal case, we'll have around 757 whole slide images for training, 191 or 192 samples for validation, and final remaining untouched dataset 105 whole slide images for testing. Next slide, please. Yeah. So this is, I created a visualization, a dataset visualization, so I can give you an idea of what exactly, the pixel density, image resolution, and tile density looks like. If you look at the right scatter plot on the presentation slide, you can see that the average pixel density, I mean, for example, the general images that you see in everyday life, they are 1024 pixels cross 1024 pixels. But as you can see here, a general whole slide image has an average pixel resolution of 1,25,000 cross 80,000 pixels. Now that is a lot, right? The everyday images that you see is only 1024 cross 1024 pixels or even lesser. Imagine having 1,25,000 cross 80,000 or 1,25,000 pixels. Now that's a lot of data. So I did a scatter plot for it. We could have smaller images as well, as you can see, but the average ones are that big in size. So the general size of this image having so much pixel resolution would be around 3 to 5 GB. Next slide, please. Yes. So we arrive at the very important part in our presentation, which is the segmentation. So now the image that you see, the fish kind of structure. Basically, this is what an image looks like when it is, sectioned, stained, and put under on a glass slide. You can see it on your right. I have written tissue slides. So these are how the glass slides look like. And as you can see, there is a lot of white area on the glass slides, and those are actually really useless for us because they don't have any useful information. The real useful information is for us is the stained area, because that's where you have your cell from the tumorous region that you can actually analyze. So you can segment out that area, and then we do the actual segmentation. The actual segmentation here is nothing but creating patches. Since you can't really process this image directly, you create patches. And you can see from the image how these small squares, when enlarged, can be, can have these cellular structures. So if you go ahead and magnify, at least 40 times, that's when you're going to see something like this coming out from such an image. I have, added some of those example patches here, and as I have given you the TCGA link, you can go ahead and you can search it now itself if you're sitting on a system, and you will see, and you can explore the website. There you will find an image viewer where you can real time expand on these images 20x, 40x, 30x, any type of magnification you want. And you can actually look at the regions expanding and the actual cellular structures. So this is how, after you segment out a whole slide image, this looks like this. So tissue slides are also called as whole slide images, just to avoid confusion. So this is how the patches are. We call these patches, and the process is called patchification. So we basically create small machine understandable tiles, which can be processed by a machine or understood by you and me if we were pathologists. At this point, a pathologist can definitely look at the tile and tell you whether it's lung adenocarcinoma or lung squamous cell carcinoma. But of course, he or she has to look at least 5 or 10 tiles like this to say what it is about, and neighboring regions as well. So, since you have seen how tiles could look like, we filter out the tiles, and we only okay, when you create tiles like this from a whole slide image, you end up creating 40,000 to 80,000 tiles per whole slide image. So from a slide looking like this, you will end up with 40,000 to 80,000 tiles. And we already discussed that we have at least 1,053 samples in our data set. Imagine having 40,000 into 1,053 samples that you need to train with. That's like a lot of data, like lot. So you cannot really train on all of those images. So we will try to narrow down our data set, right? So what we come up with a threshold, tissue coverage threshold, where we are trying to only retain the tiles which have 80% or more information. So you can look at the right, the retained tiles and the discarded tiles. You can look at the difference very clearly. Those tiles which have information or lesser than 80% or no information are discarded, and only those with more than 80% are retained. So this retained set will form the data set now. So we have boiled, so these are around 10 to 20,000. We have boiled down from 40 to 80,000 to 10,000 to 20,000. So we are at approximately 15 to 20% of the data set now. We have already removed the remaining 80% noise that we had in our data set. Next slide, please. Okay. Now you had a look at what whole slide image looks like. I have attached more examples of it to give you an idea. It could be of any shape, because it's basically a tissue area sectioned. So it could come in all these shapes. And interestingly, in all these staining colors as well. So it's just not, one color. For example, you put more of the stain, you will have a darker shade. If you put some lighter stain, or you put some light staining or less amount of staining, you will have lighter shade. So that's how even within the data set, you're going to have so much disparity. I have attached 3 types of staining: optimal staining, hypochromatic staining, and hyperchromatic staining. It's basically, optimal staining is actually the right amount of staining. So we use something known as H&E, hematoxylin and niacin staining in our case. There are multiple other types of staining that are used. Please look it up when you're free and if you're interested. So in that case, you can see how the image tiles look like. if there is optimal staining, if there is hypochromatic in the sense lesser amount of staining is applied or less stain is applied to the whole slide image, and hyperchromatic staining, more stain is applied and you can see darker tiles. This happens because say multiple people are working on creating the slides and creating the samples. So if you and me were also to be put together in a lab and were asked to make these slides, there's definitely going to be some difference in the way we stain the slides, right? You could maybe put more stain into the slide and it could look darker, and I might actually put lesser stain and it might look lighter, and that actually causes disparity in the way the tiles could be viewed, because staining is used to highlight the cell and the cellular structure. Basically, without that you cannot look at anything, it's absolutely blank. So if you apply more, the actual the cellular structures are blurred because of more staining. So optimal staining is the right amount of staining. And our machine doesn't understand all of this, the disparity in the amount of staining that could happen. So we normalize this staining. There are multiple ways that are already present. You can also look that up, which is also goes by the name stain normalization, which is used to standardize color appearance across samples. There are many, for example, Reinhardt stain normalization, Vahadin stain normalization, Masenko stain normalization. Then there's new one here, Johnson and Johnson, some stain normalization that I was looking about the other day. So these are used to normalize these stains. So now you won't have all these different color stains. You will have one standard color structure, and this helps our machine or model that we are going to use to easily understand. It's already given the large amount of data. We don't want to confuse our model with, again, different types of colors and staining as well. So yeah, that's why we use staining, no, stain normalization. Next slide, please. Okay. Many of you would have already heard about augmentation techniques, and you would have also been using it in your data. When is data augmentation used? The answer is when you have lesser data or you want to induce your real time cases in your data, which maybe you have a synthetic data and you want to have some real world cases induced in it. That's when you use data augmentation. And similar goes for medical data as well. We are using data augment. You might ask, why are you using data augmentation? You already have 10,000 to 20,000 tiles per whole slide image, and you have 1,000 of whole slide images. What's the need? But if you can remember, we discarded some tiles, and that's like 70 to 80% of the tiles which we discarded. So we'll definitely have to do something to, induce those kind of similarities in our data set, right? So for that, we use augmentation. So this way, we'll have limited extension of data set, and we are not going to end up with a million tiles to train with. So we use these augmentation strategies, as you can see. These are a combination of photometric and geometric augmentations. We use horizontal, vertical, flip, random rotation, affine transformations. So these are your geometric augmentation techniques. Then we use Gaussian noise, Gaussian blur, color, clahe, all of these to enhance local contrast and mimic mild defocus or out of plane scanning artifacts. So these, we use this together in sync to actually mimic a real world changes in or real world cases in our data set without actually having to use the entire data set. Next slide, please. Okay, so I just talked about the data augmentation techniques in the last slide. And here I have attached an example or illustration of how geometric and photometric augmentations can work together and how it can actually end up giving you real world scenarios, mimic the real world scenarios. For example, I've attached the original image and I have applied geometric augmentation, photometric augmentation, and how the resulting image looks like. We add something known as course dropout as well. So the black box that you see is course dropout. We don't give the end. I mean, if you give the entire information to your model, you also know something known as overfitting, right? So the model will learn and just simply remember, okay, this is how lung adenocarcinoma looks like. I'll predict it is lung adenocarcinoma. But there are always exceptions in medical health, if you know. So it's imperative that we provide these course dropouts so the model can guess what it could be. So in a case where there could be distortion in this area, the model implicitly knows that this is just a distortion. It's not an abnormal case and it knows how to predict properly. So augmentations are very necessary in any data set. Please, if you're using any AI ML data set, please do some augmentation to it. It's really, really helpful. Next slide, please. Okay. So this is one of the architecture we proposed. You can look it up in my paper as well. It goes by the same name. So most of you would have already heard about, efficient net data structure. Deep learning model, I'm sure. So I will give you a basic context what it is or what it does. So you've heard about convolutional neural networks, which are used to process images. We have text-based models, for example, BERT, and other transformers, which you use for, processing textual data. You have machine learning models to predict and for classification and regression analysis. You have those models. But when it comes to image data, you cannot use the same classification regression models in the machine learning that you have used before. For the categorical or numerical data, these type of images are different, and if you have time, please understand and look into convolutional neural networks. They are a fascinating topic, and the way a machine actually learns to understand what an image looks like and and predict, given an image it is actually a cat or a dog, is really, really interesting. You should look it up. So in the figure that I've shown here, which is a proposed architecture eGloconnect, I have used efficient net architecture, which forms the baseline or the foundational feature extractor. So now that we have our image data set pre-processed and ready for training, we will need a pre-trained model, right? So when I say a pre-trained model, I mean this model, efficient net. You have millions of models available in the market which are pre-trained. And pre-trained on what exactly? That's known something as an ImageNet dataset. Please Google it. And you can see that it has 10,000 to 100,000 images, and it has images of random things like a dog, a cat, a tree, a table, a bike, very random images. So it will teach you how to recognize between these generalized objects. But when it comes to understanding cell morphology, it's not enough, right? You cannot really see, learn from, for example, you have an image. You now understand what a cat looks like, what a dog looks like. But if I suddenly give you a cell structure, will you be able to understand what that is? No, right? Same goes for the machine learning model or deep learning model as well. It will not comprehend exactly what exactly is the model doing or how is it learning. It can help you maybe from the cat and dog to understand, okay, this is circular in nature, this is red, this is pink, there might be some difference because of the color. You can understand only those basic things, but if you know how to understand, differentiate between a cat or dog, you can definitely never say what a cell structure is doing, or the images that you saw, you cannot really classify based on what you understood. Same goes for the deep learning model as well. So here, when we use efficient net, which is a deep convolutional neural network, it is known for compound scaling strategy, where the network's depth, width, and input resolution are scaled and balanced to achieve both high accuracy and computational efficiency. This is one of the best deep learning models, which are also lightweight, and they have an embedded attention mechanism, which is a very beautiful mechanism. The word attention goes, what it says is focus where it is needed to. For example, if there is an image of a cat, okay, sitting in some grass, and there is like a scenery behind, and I ask you to classify whether it's a cat or dog, you have to focus on the cat, right? You won't focus on the grass, the sun, the wind, all of that. Yeah, exactly. The attention mechanism, it actually focuses on the cat pixels. Yeah, so that is what attention mechanism does. So what we do is, efficient net is trained on all of these cat dog images, which is not enough. So we add an attention mechanism to be able to understand. First of all, we train with our images as well, but to also, enhance the precision of what our model understands, we also add something known as global context attention. I have added one of the blocks explaining that. You definitely have the slides. You can look at what each of these components does. Due to time constraints, I'll not be going into the details of this, but this is known as global context attention, and we integrate this module into the architecture, positioned right before the classification head. It extends beyond local receptive fields by modeling long-range dependencies. So what long-range dependencies is, just understanding the cell structure is not enough. You also need to look at the neighboring regions to understand the relationship between various cells. There could be some disformation or some kind of indication that it's malignant and not your regular cell structure. So you also have to look at that. That is known as long range dependencies, which will allow for the network to encode global spatial relationships and contextual information. Okay, so this is our proposed model, and we have used this for training. You can look at an overview of this. So we have our data set, we have our model. Now we need a learning procedure. So I discussed with you already that we have only the slide level classification labels. The tiles that you saw, small squares, those we don't have those classification labels. So now we have 10,000 to 20,000 tiles coming out from a whole slide image, and we do not know anything about their classification labels. This is because not all parts of the tiles are benign or malignant. Only 15 to 20% of the tiles are actually malignant, which hint at the classification label. Rest are all benign. So you'll never know which part of the tile is actually benign or malignant or hinting at a subtype of lung cancer. And to actually get those annotated, if you have like, how much would it be? 10,000, 20,000 into 1053. It's almost like 1 lakh pixels, right? So 1 lakh images. So if you actually go to a pathologist and ask him or her to sit and annotate all of these images, they are actually going to say no, and it's not really feasible, right? So there is this term weekly supervised learning. We use this for training. What it does is it alleviates annotation burden. It's nothing but one-on-one mapping between your whole slide image classification label and patch level classification label. So nothing but whatever tiles we are getting out of the whole slide image, we are going to give them the same classification label as that of the entire slide. And now you might have questions here. You just said that only 15 to 20% of the slides is actually, malignant, and you're actually naming all of the slides that you obtained with the same slide level classification. So how is that going to work? Because 80% is benign. So yeah, I actually said, we are filtering out the high tissue coverage areas. And in histopathology, it's a proven fact that, higher cell density, the more the region is stained and more the cells that it has, the higher chances of proliferation and malignancy. So based on that fact, we are actually retaining only 15 to 20% of the entire region, and we are naming it, or we are giving it the same classification label as that of the slide level classification label, right? Which means we are kind of in tune with what the medical fact is, and we definitely would be naming some of the tiles as wrongly classified. That is, of course, there. That's the name. That is why the name is weekly supervised. Supervised because we are giving classification label to every region, but weekly because not all of the classification labels that we provided is correct. Okay. Now we train on these tiles that we obtained, and we'll get the classification label for these tiles. So we'll need to aggregate them, right, to get the whole slide level classification. Yeah, so exactly. So we have majority voting, probability averaging, and weighted voting. So majority voting is the simplest thing. Basically, say I had 100 tiles from a whole slide image, and 100 out of, say 51 tiles of those 100 tiles say it's lung adenocarcinoma, I'll declare that the entire whole slide image is lung adenocarcinoma. This is what majority voting does. Similarly, probability averaging. Probability averaging is actually based on confidence scores. So if you know, you would actually, take out the softmax classification probabilities and average them. And whatever you get, if it is beyond a threshold, you will classify the slide level classification. And weighted voting is again based on confidence. Of course, you can look it up. I just gave you the simplest one, which is majority voting, which we also used in our case. Next slide, please.
Go first. Is it as quick as possible? Time is approaching.
Yeah, sure. Next
Is
slide.
over, no?
Oh, next slide.
No,
Okay.
no.
No, it's not. Okay, so I gave you the entire steps in the process, right? So let's have a quick overview of, the uh, steps involved in processing such huge data. So as you can see, we take the whole slide image, we segment it, we create patches which look like these. I already showed you those. And we perform sampling. That is, we took out only those patches with more than 80% information. That is, which are very more stained and not those with lesser stained areas. And I did some stain normalization and augmentation, as you can see. You can follow the arrows. That will be easier for you. Then in the third step, we performed. Then we used weekly supervised learning and our proposed architecture, eGloconet in the fourth step for model training. Then we obtain classification labels in the fifth step, as you can see, and we aggregate them. Like all of these will be aggregated to get one slide level classification, as you can see in the last step. So we have six steps in the process. And if you're interested, if the slides are provided, you can look at the results of it. And
Next slide, shall
we,
I go?
yeah, please, you can go to the next slide. I'll just. Yeah, so this is a table comparing our model with other baseline architectures. We have used very recent vision transformers and also very lightweight. And since the data set is already heavy, we always focus on having, very lightweight models so that it doesn't impact your CPU or GPU when you're training or testing it. So we have compared our model with very state-of-the-art baseline models, and you can see our model has outperformed all of them. You can go through the results and classification metrics at your leisure. Next slide, please.
Able to see or not?
Yeah, I can see now. Okay, so before I conclude my case study or presentation, what I would like to say is our eGloconet or our proposed architecture, gains an excellent AUC or classification accuracy. As you can see, it is 0.9648. It's way beyond what medical or clinical industry actually expects it to be, and we have reached that percentage. And you'll be happy to know that we are collaborating with AIMS Bhopal as well. So we are working on their data, and we are getting such great accuracy that it will be deployable in the medical industry soon. And the most important part is we are using only 15 to 20%. And in our case, we have boiled it down more. Due to time constraints, I could not explain the sampling strategy, but we used only 7 to 10% of the entire whole slide image. The size of this data set was around 1 TB. So when I say 7 to 10%, we really, really boiled down the data set. I think it was around 80 to 90 GB that we trained on instead of the 1 TB. And we are getting such excellent results that it is efficient and also scalable and easily deployable anywhere. Um, so, uh, like any other research, this research definitely has its own limitations, and there are many future directions that could be explored. You could use CNN-based architectures, vision transformers, and now we have vision language models as well. You can use clinical history of the patients to go ahead and explore more about it. So yeah, that is it from my side. I would take the questions from you, if any, at the end of the presentation. And with that, I'll conclude this study. Thank you.
Please, any questions, please?
Yes, I'll be happy to take any questions if you have.
Yes, let me ask.
One question is there in the chat box. What is the best method or tool of converting unstructured data to structured data for medical field data? Sir, am I audible?
Yes,
Yeah,
yes,
yeah.
you're audible. Okay, so if you have that question for me, I already showed you a very good example of how you can convert your unstructured data to structured data. About the processing steps that you apply, depending on the field that you're using. For example, we used medical images, and I showed you how the image could be vast and unstructured and all over the place. You can actually go ahead and use various pre-processing techniques. For example, in our case, segmentation, stain normalization, data augmentation, and all of those techniques you can use to, structure the data in the medical field. There are multiple other types of data, for example, textual or sentimental data, sentiment-based data, which also Sir talked about in his earlier slides. You can also process that. What that does is, if you have, for example, clinical data, for example, the patient history, whether he's smoking, what age is he, what race is him, if he has some clinical history of his parents or anybody having that kind of disease, you can actually also go ahead and pre-process that data. That is going to be unstructured data, and you can look up the various ways in which text data is cleaned and pre-processed. So those steps will come in when you want to convert unstructured data to structured data. It's very subjective and depends on the type of data you're using. Yeah, I would say that.
Just see the messages are coming.
Yes, yes, I'm looking at it. So we have a question from Mili Panigrahi, ma'am. So she says, can we apply multimodal images for AI modeling for seizure prediction? Yes, of course. Why not? It's medical data. And for seizure, I mean, you can come up on the chat and talk to me. For seizure prediction, I'm not sure what kind of data you're using. Is it medical data? Is it biosignal processing data or clinical data? So, what type of images do you have? You can definitely use 2 to 3 type of images. You can club together the features that you extract out of them, and then you can use an encoder. You might have heard about generative AI models, GAN. You can use a GAN encoder, encode all of those feature vectors together, and definitely you can learn from it and predict. Yes, you can apply multimodal images. The next question is from Yatendra Pathak, sir. He says, my field is NLP in cardiovascular disease. Any suggestions? Yes, of course, you can connect with me offline, but NLP in cardiovascular disease, I'm thinking it would be more about the patient information. If you could provide me more details about what exactly the data says, I can help you more. You can definitely look up BERT models for textual and sentiment analysis. And there are other models as well. I cannot recall them at this moment, but there are other uh, models trained on medical data itself. So it will be helpful if you can look up those models. They are trained on clinical data, so textual data. They'll be more helpful because they're already pre-trained. And then Mrs. Lucky Harichandan says, is there any procedure to getting medical data? Okay, so I already told you there are multiple data sets available online, right? They are freely available and only, I mean, available for the research industry to use. The one that I've used is publicly available data set. You can go ahead and use it if you would like. There are multiple such data sets for breast cancer. I mean, this data set, TCGA, has 18 or 32 types of cancer data. There's vast data available on the internet, please. You can
No,
definitely go ahead and look for it.
1 minute, Asurya. Tell
Yes.
the sites where data is available so that they can Google it
Yeah,
and find.
if they just go ahead and type TCGA lung cancer, they'll find the data sets. It's that simple. Just go ahead and look for it. If you want any specific type of data, you can definitely ping me here. I can, link the URL in the chat. If you want, I'll link the TCGA URL right away. You can definitely go ahead and look at it.
So any other questions? Yeah.
Can you write it down in the chat box from where they
Yeah,
will
yeah.
get the?
Just
Yeah,
check
yeah.
it
I'm
out.
adding the URL to the chat.
Yes.
Yeah.
And now another question is from my side is that you have
Yes.
taken 5-fold cross validation.
Right.
This 5-fold cross validation is the standard validation or what? Why you have taken this 5-fold cross validation?
Okay,
Why
so
not it is less than 5? Why not it is not more than 5?
Okay, so this is also, you might have heard something about grid search cross validation. There is K-fold cross validation strategy. You could go for two-fold, three-fold, eight-fold, 10-fold, 15-fold. It does not matter. So five-fold specifically because we test two, three, four, seven, and five-fold turned out to be the most optimal in a way. The CPU or the computational complexity was the computational complexity we were getting, the accuracy we were getting, everything was very optimal with five-fold cross validation. So we took it. You can definitely go ahead and test
Then
with seven-fold cross validation, 10-fold cross validation as well. But considering our data, 10-fold cross validation was too much for us. So we restricted it to five-fold cross validation.
It means one trifold is the optimal cross validation. Can I say that?
No, you cannot say it is actually the optimal one. It is very subjective to the data you're using. For example, I have seen some works from Professor Mohanty's research, and he has used 10-fold cross validation, and they gave excellent results. You could actually look up his research, and you will know. So, what I would say is please go ahead and play around with the data, see what works out. If you're interested, start with 5, 10, 15, go like that. Take those numbers, see how much time it is taking for you to actually arrive at the results. Too much time is also not good, or very less time, but not a good accuracy is also not good enough for us. So look for what is optimal for you. What do you define as optimal? Some amount, some threshold of accuracy, and some threshold time for training, right? So consider those, and then you can define what kind of validation strategy you would like to use. It's all subjective.
Okay.
Yeah.
Any
Anyone?
other question? Participants? Participants, if you have any query to Aishwarya ma'am or Ramakanta sir, you directly write it down in the chat box.
Yes, you can also write it on the in the chat, and you can also discuss if you would like. You can unmute yourself. That'll be better. So I've provided the links to you in the chat. The second link will take you to all the cancer types. I think they have listed around how many are there?
around
Some
65.
Yeah, not 65, some 10 to 18 or 20. Those
No. Initially they joined 65 people. Later they might have left. And now at present 44 people are there.
oh, I'm okay. It's not talking.
Okay. So lastly, I want to say very plain words. What is the roadmap to learn big data technology? First of all, if you are starting research or you want to register or you want to go ahead, those are middle level is fine. What she told is fine. But those are going to start. I want to give a roadmap to start research in the field of big data analytics by using any type of problem. First of all, you learn Python, advanced Python, Python for machine learning. And these results, another language also, they are called R language. Now, if you compare this R language and Python, R gives more accurate compared to Python if research is concerned. So better
I
to go
agree.
for, yes, better to go for research R programming and do research and paper will be accepted comparatively better. Number two, for big data analytics, you should know very well first SQL, then MySQL, and go for PostgreSQL because this is required to have a number of queries for big data analytics. Next, base levels you start with what is called SDFC, that is called Hadoop, then followed by MapReduce techniques. Then you follow Apache Spark, and no SQL is needed because you want to create a very big database. And in addition to this, for visualizing your results, you must learn not Power BI, Tableau. Tableau is now used most of the cases. And learn well machine learning techniques, basic level techniques, then learn deep learning like CNN, then Many more techniques are now generative AI, then extreme learning machine, then kernel-based support machine, not linear support machines, random forest, you know very well. And in addition to this, if you're not getting better results, go for ensemble method by adding 2 or 3 methods like evolutionary component methods like genetic programming, ant colony optimization techniques, firebird optimization techniques, or you will go for swarm optimization techniques. Those techniques are giving very good results nowadays. You just combine 2 to 3 techniques, make an ensemble or hybrid method that will give you very good results. Don't go for a very big data set, go for a small data set. And if you are going for hybrid type things, getting published paper in good journals will be very easy. So with this, I'm just ending my session. Thank you very much. Thank you all.
Thank you so much, sir. Respected Manti sir, Aishana ma'am. We extend our sincere thanks to both of you for your highly informative session on big data in healthcare, tools, ethics, and insights. Your detailed explanation of data analytics framework, ethical consideration, and the transformative role of big data has greatly benefited all participants. We truly appreciate your valuable time and meaningful contribution to our everyday.